Hi there, my name is Dr. Raihan Falari, and in this class we're going to be discussing about the cell and its organelles. All cells are generally of two categories. They are either eukaryotic or prokaryotic. For the cells that are prokaryotic, their examples are bacteria, and these are the kind of cells in which DNAs the deoxyribonucleic acid that is responsible for the hereditary transmission from one generation to the other is located within a membrane separating those DNA from the rest of the cell. That's the thing with prokaryotic cell. And usually in this kind of cells, cell walls are always present in all types of prokaryotic cells. However, in eukaryotic cells, the chromosomes which are responsible, which contain this DNA segment, they are located within the nucleus. And the nucleus is just one of the other membrane-bounded organelles. So cell walls are also present in just the fungi and the plant type of eukaryotic cells. In other words, eukaryotic cells could be plant, could be fungi, could also be animal cells. So for animal cells, they usually do not have cell walls, but for plants and fungi, they are the only types of eukaryotic cells that have cell walls. Generally, eukaryotic cells, and like I said earlier, they contain membrane-bound organelles. So, like I mentioned earlier, here is an illustration of prokaryotes and eukaryotes. Prokaryotes, here is bacteria cell as an example of it. It usually has no membrane-bound organelles. While for eukaryotes, it includes animal cells and it includes plant cells. These generally always have organelles within them. And that's our concentration in this class majorly. So all cells generally are surrounded by a plasma membrane, and that's the membrane that is bordering the entire cell. They always have a cytosol, that's the fluidy content in which all of the organelles are submerged. So we call it cytosol because it's a semi-fluid substance within the membrane, and by that way, a cytoplasm is combination of the cytosol and the organelles. So the cytosol is the fluidy substance in which the organelles are submerged. So when you combine the cytosol with the organelles, that can be referred to as the cytoplasm. So all cells contain chromosomes, and the chromosomes are the ones that carry the genes on a double helix strand known as the DNA. All cells tend to have ribosomes, right? And these are tiny organelles that make protein using instructions that are contained in the genes. So this is a way to let you understand the cell size comparison between animal cells and bacterial cells. This is like a ratio 1 to 1 million uh, meter, okay, or micron. So this is more or less the diameter of a human hair, which corresponds to 20, approximately 20 microns. So here, most bacteria are generally 1 to 10 microns, while most eukaryotic cells would generally measure from 10 to 100 microns. Here is another typical illustration of that. So one thing you should, however, have at the back of your mind is that the cell surface area tends to increase, and as it increases, the total volume of the cell tends to remain constant. The cell surface area may increase, but the total volume of the cell may remain constant. For example, this is a cell. Yeah, let's assume this is a cell, and here is another block of many cells that have been subdivided into this chunk. or this little box of cell. So the total surface area that is covered by this little box here, which is calculated by the measurement of the height with the width and then the side. Okay, here, if it's 6, right? If it's 6 here, there are a whole number of this one in this bigger box. And that increases. So if the volume here, if the total surface area is 6, over here, the total surface area is 150 because the size has increased, the surface area has increased, and then the volume, total volume is going to be 125. The total volume of this little one here is 1, okay? And then the surface to volume ratio, okay, is still the same 6 here. While for this that has increased only in volume, or let's say in size, has increased in its volume as well. Okay, it has increased in surface area by 150. It has therefore increased in its volume by 125. But look at the surface to volume ratio. It's just 1:2. Okay, it's just 1.2 rather. While in this case here, this same volume has been divided into sub chunks, right? And what has changed here? The volume remains constant. So as the cell increases in surface area, a cell may increase in surface area while the volume would remain constant. So that's what you have here. The total surface area is the only thing that has increased in this case. So because the volume here and the volume here is still the same. So the surface area of a cell may increase based on the amount of functionalities it needs to have, based on the number of organelles it needs to have to be able to perform several functions. It doesn't necessarily mean the volume of that cell has to increase. So having an increased surface area enables the cell to be able to compartmentalize, to be able to divide itself into compartments while enabling its functionality through those organelles that reside in those different compartments. So this is generally how the cells behave. So despite the subdivisions you have in this box, you would notice that each of the chunks remains the same in volume, and therefore the total volume remains the same with this total volume. And that's why you have these here. Okay, but when you look at the surface area, it has increased, and that gives us 750 as compared to 150 here. While here, you would notice that the surface to volume ratio, the surface area to volume ratio, is 6. So why do we study cells? We understand that cells aggregate to form tissues, and tissues aggregate to form organs, and then organs come to make up the entire system of the body. So the bodies are made up of cells, and cells do all the works that is expected of life. And when all these cells come together, it's only then that you can have an entire individual, regardless of what type of individual it is, whether it's a lower animal or a higher animal. So why organelles? Organelles are specialized structures that perform specialized functions. Okay, so whatever the function it is, it enables the animal to live and enjoy and be able to function optimally. So such structures could include the cilia or the flagella, which helps to move. Organelles also go through compartmentalization, like I illustrated in the previous slide. And this is a way of partitioning them into compartments, creating different local environments, creating distinct and incompatible functions. Organelles, so by virtue of this, they show that they include membranes, and these membranes help to enable chemical reactions. So there are places where chemical reactions occurs. There are places where the receptors are located. So they enable unique combination of lipids and proteins amongst that. So organelles are very diverse in their functions. So cells generally have three main functions. So the various organelles that perform these functions for the cells. So one of them is that the cells generally they make energy, cells make proteins, and cells make more cells. They make more energy in the sense that you need energy for all activities in the body, right? And the body is broken down back to the level of cells. So for the entire body to function, it means the cells must be functioning optimally. And for them to do that, they must be able to make energy. So they need the energy to also clean up waste produced while making the energy. The cells make proteins because without proteins, there will be no work possible during the in the cell and in the entire body. And then the cells need to continue to reproduce themselves, whether at times of injury or for the purpose of growing from small individual to being a larger or bigger individual. The cells need to be able to reproduce themselves, multiply, replicate themselves, and replace themselves when there are damages. And there are times when cells need to undergo renewal, where older cells need to shed off or go away, while some new cells take the place of these. So our organelles do all these jobs for the cell. So organelles do all these work, and each structure, each organelle has a duty that it's performing within the cell. So as regards the making of energy, energy is the fuel that helps for daily life and growth, and therefore the cells must take in food, they must digest the food, must take in oxygen, they must make ATP for energy, and the cells must work to remove waste. So it needs energy for all these. So what organelles do this? They include the cell membrane itself, they include the lysosome, they include the vacuoles and vesicles, and they include the mitochondria. As regards the cell membrane, they help to separate the cells. Uh, they separate the inside environment, the inner environment of the cell from the outside environment. They control what enters the cell and what leaves the cell. They recognize the signals from other cells. They help the cells communicate with other cells. So it's composed of a phospholipid bilayer, like you may have been introduced to in more biochemistry-oriented lectures. So they contain receptors on these membranes that help the cells communicate, receive impulses, and also export its, uh, its responses. Lysosomes, as displayed here, so they help in digesting things, digesting food that the cells need to be able to function. So these are also needed to make energy. They also help in cleaning up and recycling already digested wastes that the cell produce. So it's made up of a membrane sac of digestive enzymes, as you'll see here. So that's another organelle within the cell. Also, you have vesicles and vacuoles, which function to help move these materials around the cell, useful materials or waste materials around the cell. They help in the storage functions of the cells, and they are generally made up of membrane sacs. So, uh, mitochondria, and the mitochondria, which whether in animal or plant cell, they function chiefly they are the most paramount energy making organelles in the cell. So they make the ATP energy from cellular respiration. It's also composed of a double membrane, which can always be identified histologically. So here is an electron microscopical view of the mitochondria, showing the inner membranes, as you can see here, and then the cristae inside. So the inner membrane is there, the outer membrane is there, then it has a matrix within it, and then you have the cristae uh, helping to further compartmentalize the inner space of the mitochondria. So you find mitochondria both in animals and in plant cells. And as for plant cells, they have an additional way of making energy, and that is through chloroplast, but that's not our focus today. So we mentioned already that the mitochondria uh, helps the cell to generate energy, especially in animal cells. So almost all eukaryotic cells have mitochondria, and there may be one very large mitochondrion or hundreds to thousands of individual mitochondria. The number of mitochondria is correlated with the aerobic metabolic activity of the particular body or of the cell or of the system in which the cell is found. So there are quite a number of cells that are expected to have a lot more mitochondria than other cells. What kind of cells are those? They include muscle cells and nerve cells. Another function, so after talking about the energy making function of the cell, another function of cell is making of proteins, like we mentioned earlier. So cells need proteins to run daily life and growth, and therefore the cells must be able to read the DNA code and then build proteins over this. So the organelles that help the cells do this include the nucleus, the ribosomes, the endoplasmic reticulum, and the Golgi apparatus. So that's also the sequence that it follows. All right, the whole process starts with the from within the nucleus, which is located within. It's the central organelle within the cell, and then from this, the ribosomes are formed, and the ribosomes get transported, and then you find them on the endoplasmic reticulum, and then they get transported through the Golgi apparatus, and then you find them in vesicles. So the organelles involved these, as has been illustrated in this image here. Control the entire functionalities in the cell. So it's the center, is the control center of the cell. It contains the DNA and houses it. It's within the nucleus that you find the chromosomes that are bearing the DNA double helical strand. So it's from the DNA that instructions for building the proteins are gotten. All right, it's the DNA that contains that. And the structure of the nucleus is such that it is bounded by a membrane. So apart from the fact that the entire cell has a membrane, the nucleus within it is also it's also bounded by the nuclear membrane. And then the nucleus further has within it something called the nucleolus. The nucleolus, that's where the ribosome is actually formed. And then within the nucleus, you have the chromosomes as well. So within the cell, you have the nucleus. Within the nucleus, you have both nucleolus and chromosomes. And there are some times when the chromosomes, where the nuclear membrane will have to degenerate for the purpose of the interface phase of cell cycle, and that will herald reproduction or replication of DNA materials. So during such times, you will find the chromosomes lying around within the cell and not being housed within the nuclear membrane. But aside those times, you would always find the chromosomes condensed within the nucleus. So yeah, ribosomes function as the protein factories, and they help to read those instructions to build proteins from the DNA. So the structure is such that you have some ribosomes always free within the cytoplasm. After they have been produced, you find them free within the cytoplasm. And you find some rather attached to the endoplasmic reticulums. So these little dots indicate or represent the ribosomes here. So they are attached to this rough endoplasmic reticulum. And the reason why they are called rough endoplasmic reticulum is because of the attachment of the ribosomes to them. While for those endoplasmic reticulums that do not have ribosomes attached to them, they are referred to as the smooth endoplasmic reticulum. So that's what we've illustrated here again. So the endoplasmic reticulum functions to work on the proteins, and then it makes membranes. So it is structured such that the RAR, that's the rough, contains attached to ribosomes, while the smooth does not have the ribosomes attached to it. So there are also some cells that are expected to have much more rough endoplasmic reticulum than other types of cells. So example of such would be cells like pancreas, which are responsible for the production of digestive enzymes. Okay, so that's a typical example of that. Yeah, so for smooth endoplasmic reticulum, it's chiefly concentrated on membrane production, as well as many other metabolic processes like synthesis of lipids, hydrolysis, amongst others. So also implicated or involved in the making of proteins is the Golgi apparatus. The Golgi apparatus, and that's what you see illustrated here. So it functions to finish and then sort and then tag the cell products that have been made, and then it conveys these products into the vesicles, which are more or less like membrane sacs. Okay, so we can see within a typical cell, even though this is a plant cell, those basic some of these things that we have mentioned are also present within the plant cell. So the third main function of cells is replication. Okay, so we have mentioned the organelles of the cell that are responsible for protein production. We have also mentioned the organelles of the cell that are responsible for energy generation. So the third main function of the cell is replication or reproduction of cells, making of more cells. So because the cells need to replace themselves to repair when there is damage, and then to be responsible for growth. So in order to do that, the cells must be willing to copy their own DNA and then make extra organelles, and then divide the new DNA and new organelles between the two new daughter cells. Okay, so you having a central cell divided into two main cells. So there are some organelles that are implicated, that are responsible for making this happen. And these include the nucleus, which we have talked about earlier, and the centrioles, the nucleus and the centrioles. So we've talked about the nucleus earlier, while as for the centrioles, their main function is to help coordinate cell division. So you find them aligning, first of all, at the same pole of the cell, and thereafter they move to opposite pole of the cell in order to attract this the formation of daughter cells from the orientation of the spindle fibers. So you find centrioles only in the animal cell, and they are structured such that you have one pair, that is two, okay, one pair of centrioles in every cell. So even the centrioles gets to be reproduced in order for synthesis of the necessary hereditary materials to happen. So the cell needs to be able to digest itself during all of this process, and that's where the lysosome functions. So uh, they bring their function to ensure that they break down the waste or things that need to be digested for the cell. They break it down, like we mentioned earlier. So lyso means to break things, and the some means body. So through the lysosome, structures that occur as polymers get digested or broken down into monomers, and then they pass this to the cytosol to become nutrients of the cell. So yeah, this is a further emphasis on how the lysosome help to digest macromolecules, and then they clean up the broken down materials from the other organelles. So they are structured in such a way that they are vesicular. They occur more like vesicles, as shown in this electron micrograph. So as vesicles of digestive enzymes, so they contain enzymes with which they help the cell to digest things. So lysosomes are synthesized by the rough endoplasmic reticulum and get transferred to so ribosomes that are synthesized from within the nucleus and get passed uh, to attach to the rough endoplasmic reticulum, get transferred through the Golgi as mentioned earlier. So that's what the lysosome does. So when cells need to die, there are times when, through feedback mechanism, there are sensors in the cell that monitor growth. They trigger self-destruction when they sense a process needy of such. So lysosomes can be used, to kill cells when they are supposed to be destroyed, right? It's part of the natural process at times. Okay, so that's what is happening in this animal you see here. Even the brown spots on leaves too, times, or maybe through virus infected plants cells, those ones also auto destruct, and even cells around it. So they auto destruct in order to wall off the virus. So lysosomal action is very important for normal living of the cell. So some cells have to die for the proper development of an organism. So this brings us to the concept of apoptosis, which is natural cell death. It's an auto-destructive process, which is normal at times, right? It is normal. So it is through the lysosomal action that things that need to be broken down gets broken open and then they get eaten off. So example is what you have in this tadpole, where the tail gets reabsorbed when it turns into a frog. Okay, so that is a natural lysosomal action and an apoptotic action. So another example that is that of the losing of web fingers. Everybody gets through that during embryological developmental process. Because at age 6, you tend to have webbed, very webbed limbs or hand plates. Okay. So if it remains like that and doesn't get separated into fingers or toes, you end up having congenital anomalies like these, where fingers are joined or toes are joined. Okay, that's because there is incomplete degeneration of those cells. There is incomplete killing of, eating off of those cells that lie between those fingers. So you would agree now that at times cells need to die or cells need to be eaten off. So those cells that are joining the two toes together or the two fingers together were not properly eaten off, or they were not properly digested by the lysosome at that point. So if these lysosomal actions, apoptotic actions do not occur at the right time in the right magnitude, it leaves some undesired outcomes, such as webbed feet or webbed fingers, like we always refer to them. Um, yeah, so that's what is referred to as syndactyly. So another basic function that some of these organelles perform is in terms of food and water storage. So other organelles within the body include things like peroxisomes, which contain enzymes that transfer, you know, uh, hydrogen to oxygen and help in making of hydrogen peroxide. This is important in detoxification of fatty acids. They help in detoxification as well as in the hydrolysis of fatty acids. And you have the peroxisome labeled here in this electron micrograph. So by this virtue, they then convert the hydrogen peroxide back into water. Cytoskeletons are also uh, organelles of the cell. They are network of fibers that support and shape the cell. They are responsible for how much of support or framework that the cell has and what the eventual shape is going to be. They're responsible for the anchorage of organelles and enzymes generally. So cytoskeleton help in cell motility because without them, the proper movement of cell will not be possible. So that's why their interaction with motor molecules is very important for the cell survival. So movement of cilia is by virtue of cytoskeleton. The movement of flagella in paramecia also by virtue of the cytoskeleton that it contains. So cytoskeletal components can slide past each other, and that is why contraction of muscles cells becomes possible. So when we talk about cytoskeleton, we'll be talking about three main fibers. So the microtubules, the microfilaments, and the intermediate filaments with their diverse peculiarities. So it's good we also talk about the cell walls because generally the cell wall is what uh, uh, encompasses the entire cell. But like we said, that is something that is not so peculiar to animal cells. Okay, even though plant cells are also examples of eukaryotic cells. But for extracellular matrices, so that's the surface. Those are the glycoproteins you have on the surface of animal cells. So they are embedded in the proteoglycan network, like is illustrated here. So you have the plasma membrane here, and then submerged within those plasma membranes are the integrin fibers, which are also associated around the microfilaments of the cytoskeleton. All of these within the cytoplasm on the inside of the cell, while on the exterior of the cell, you'll be seeing the collagen fibers and their interactions with all the polysaccharides, the proteoglycans molecules, amongst all of these. So these are the components of the extracellular matrix. That's the things you find on the exterior of the cells. So we have seen those glycoproteins there and how they bind to the receptor molecules of the integrins. They attach the extracellular matrix to the plasma membrane in order to coordinate any changes. So the extracellular matrix is able to coordinate the behavior of the cell. Whatever happens in the ECM dictates to the cell what it should do. Also important to mention is the intercellular junction. The intercellular junction. So which is if we are talking about the plant cell walls, we're talking about the plasmodesmata. And this may be important to people, to drug developers who may have to make use of plants in their development of drugs. That's why it's important to take note of the functionalities or the anatomy of the plant cell at times. So it's what allows the cytosol, the water, the proteins, RNA, the small solids to pass between one cell and the other. They find cell intercellular junctions within animal cells as well. So here is a typical composition of the cell layers. So the plasma membrane is lining the channels of the cells. In a typical animal cell, you have it as either tight junctions, desmosomes, or gap junctions. In the case of tight junctions, we're talking about membranes of neighboring cells being fused, and this will prevent the extracellular fluid from leaking between one cell to the other. So those are tight junctions, like the name suggests. Okay, those are tight junctions, like the name suggests. While for desmosomes, in this case, they are otherwise known as anchoring junctions, and what they do is to fasten the cell into strong sheets, so more or less like rivets. But this will still allow some as necessary. While for gap junctions, like the name suggests, so they are more or less like communicating junctions, and this provides cytoplasmic channel between adjacent cells. So it allows small ions and molecules to pass, and it allows the flow of ions in order to coordinate cell functions. So on this other side, you can appreciate these different types of intercellular junctions within the animal cell. So here is a good place for us to pause today again, and you would remember this particular slide and this particular picture of a cell. By now, you understand the basis of all of these organelles. So I hope this has introduced you to the cell and its components, and then we'll be talking about other things that have to do with cell and as well as the tissue, the epithelial tissues, and then the nervous tissues in the subsequent class. Let me know questions you have, and all the very best as we see in the next class. Bye.